So thank you so much, Ariela, for having me and this opportunity to share with you a little bit of my work. The single most important thing that I want to share with you today is that the integration of highly multi-omic dimensional data with computational system biology provides a nice, really methodology to elucidate new mechanisms driving diseases. So I'm gonna show you the potential of genomic scale metabolic models to advance in the understanding of molecular mechanisms driving diseases. Next slide, thank you. So I'm gonna talk to you about biology, not computer science, but I'm gonna show you how the systems biology really have the potential and the effect for having a big impact on how we understand biology. But for those of you from the math or computer science background, I might have called this talk just system biology. So let's start with apparently is a simple disease. It's a disease called Huntington. And you don't have to be a neurobiologist to be able to tell the difference between the brain of a patient who does not suffer from having problems on the right and the brain of a patient with Huntington's on the left. With a tremendous loss of brain structure. Now, the origin of the disease mutation in a single gene is a single gene. And so you will think that this will be a case where if we understand how mutation causes disease, therefore we'll be able to figure out how to cure the disease. But this mutation has been known for 20 years and was not led to an effective cure or an effective treatment. And that's because when a single gene is mutated, affects a lot of biological process. So the traditional approach in the study of these diseases are not getting us to the kind of answers that we need. Historically, biology and biochemistry have a reductionist framework where individual components are carefully enumerated and usually studied independently. So the next one, and the next one, sorry. Okay, so the emergence of omics data over the past decade has led to a wave of new technologies that allow the measurement of almost all cellular components. So next generation DNA sequence can elucidate the genome sequence, while transcription, proteomic, and metabolomic profiling are becoming common technologies that measure mRNA, proteins, and small metabolites under specific conditions. So the generation of omics data is becoming easy and cost efficient, but the interpretation and physiological assessment of the massive amounts of data has been a daunting challenge. Next one, please. So in system biology, what we are trying to do is make a whole series of very systematic measurements of what goes on the cell. Pretty much any property we can effectively measure. Then we use computational approaches to build up relatively current views of what all those data mean and allow us to understand how signals transfer through different parts of the cell and where we might be able to go and intervene and cure diseases. Next. So reconstructing a genome scale metabolic network involves determining and formally organizing the known biochemical transformation in a cell or organism. And the biochemical network can be studied or can be converted into a mathematical format for predictive simulation using constraint-based modeling and flux balance analysis. Next. Since the reconstruction of age influence in 1999, there has been a fast growth in both the number of reconstructions and their uses. Today, there are numerous prokaryotic and eukaryotic genome scale model reconstructions of major pathogens, such as tuberculosis, aureus, or pylori, and industrially relevant bacteria as well, such as E. coli, cerevisiae, and in 2007, the genome scale reconstruction of the global human metabolic network recon was complete. Next. So RECON1 is a global human metabolic network. So omics data, such as transcriptomic proteomics, were integrated with RECON1 to determine the cell-specific reaction needed to build a cell-specific model. Algorithm approach have been developed to quickly build cells and tissue-specific models. They have been achieved the most complete and best annotated consensus of human metabolic reconstruction. This gene is available and thereby increasing the ability of the resource to provide novel insights into normal and disease states in humans. The last RECON 3D, includes information of 3,200 open reading frames that encode metabolic enzymes catalyzing 13,500 reactions or 4,100 unique metabolites and protein structure information from protein data bank and metabolite structures. It is capable of performing flux balance analysis to integrate and interpret a variety of emerging data types, including linking mutation, identified for human variation data, or cancer genome atlas, relationship between genes, the proteins that they encode, and the reactions that proteins catalyze, such as GPR reactions, are now described in the context of their specific 3D configurations, interaction, and properties. New data types include representative structural domains of proteins, metabolite structures, along with their conserved moieties, and also iron-iron atoms mappings. These genomes models of human metabolites, RECON1 and RECON2, has been used to predict biomarkers of inborn errors of metabolisms, to identify drug targets and off-target drug effects, to study cancer metabolism, and to improve understanding of microbial interactions with the host organisms. Furthermore, while human metabolic reconstructions are of obvious utility into the medical and pharmacological files, it is worth noting that reconstructions of mammalian biochemical networks also act as blueprints for modeling systems of biochemical significance. So we are going to see in the next slide some nice application that we are focused on. The next. So the growing importance of astrocyte has led to a the growing importance of astrocyte in the field of neuroscience has led to a greater number of computational models devoted to the study of astrocyte function and their metabolic interaction with neurons. The modeling of these interactions demands a combined understanding of brain physiology and the development of computational frameworks based on genome scale reconstructions, system biology, dynamic and steady state models. So this computational approach have helped to highlight the neuroprotective mechanism triggered by astrocyte and other glial cells, both under normal or during neurodegenerative process. Next. So we reconstructed the astrocyte-specific model using omics data to simulate normal, healthy, and ischemic condition by decreasing oxygen consumption. This model contains 5,000 metabolites and 5,600 reactions, which are found in 8 cellular compartments. The model also includes several metabolic reactions of amino acid, lipids, carbohydrates, purines, vitamins, and cofactors. Also, we include energy metabolism and glutamate cycle. Finally, we identify significant changes in some antioxidant enzymes associated with the prevention of brain injury during ischemia. The next. Then this model was used to test the effects of tibolone, which is a synthetic neurosteroid. And we do that under metabolic inflammation by palmitic acid-induced toxicity and the effects of tibolone treatment. Metabolic simulations were made from two different metabolic scenarios, healthy scenario and an induced inflammation by palmitate scenario. We found out that the model and also the results are similar to those previously reported from astrocyte models and also similar to previous experimental studies. Moreover, this model allows us to identify the metabolic changes between the two metabolic scenarios and the responses to tibolone. As well, also we found protective effects against inflammation, oxidative stress, and metabolic dysregulation. The big picture is that the genome scale model of the human astrocyte can be further explored in the study of metabolic alterations during other brain pathologies. The next. A central challenge in the development of system biology is the integration of omics data to generate predictive computational models, because these technologies have provided quantitative data for thousands of cellular components across a variety of scales. Given the recent success with rising genome scale metabolic network reconstruction, such a platform for integrating omics data, we are focusing our efforts on multi-omics data integration. We have been doing this work by obtaining transcriptomic, proteomic, and metabolomic profiles from human astrocyte and integrating them into a genomic scale model of human astrocyte to generate phenotypically accurate and predictive computational models. The next. We are also working on the identification of networks surrounding drug targets, and we use this data also to identify novel targets in the same pathways or to identify common drugs resistant mechanisms. One of our goals is to use novel and non-chemical compounds as molecular tools to further understand cellular mechanism underlying some neurodegenerative diseases. And now we show the leading players in the development of and progression of these pathologies are suitable targets for polypharmacological manipulation from multi-omic data. The next. In an effort to make the mentioned omics data more accessible to researchers and students around the world, we create the Astrocyte Neuron Simulation Environment Platform, ANSIP, to operate those metabolic models developed with omics data, obtained through an easy-to-use and accessible interface. The platform makes possible to perform flux balance analysis, flux variability analysis, and robustness analysis. Next. So, ANSIP attempts to reduce the learning curve for the specific community members interested in getting involved in the study of neurodegenerative diseases through a simplified environment that does not require advanced knowledge of programming to perform simulation over the neuron astrocyte metabolic models. From what we know, ANSEP is the first web platform that allows users a greater understanding of neurodegenerative process in an holistic approach, making things easier to researchers the tasks of generating data to analyze. Next. In short, metabolic reconstruction contains the metabolites and stoichiometry of the known reaction that comprise a specific metabolic function in an organism and genes associated with the catalytic activity required for each reaction. The metabolic models available in the platform are astrocyte and neurons. So here we have E. coli just for doing tests. The format of these models is an XML-based format. The next. One of the features of ANSEP is that it makes easy to perform different analyses, as there is no need for users to write a code. In the analysis module, users can select the metabolic model and configure the parameters of the analysis. In ANSEP, each model has 2 pages. One used in the case that the user has analysis expression data to attach to the model, another to manually set the boundaries of the reactions. Each analysis page contains the options to modify the objective functions and to return and to perform a flux balance analysis, flux variability analysis, and robustness analysis in separate panels. Next. Once an analysis is performed, the user can export the results as a plain text file and save the results into a personal space of the user as a JSON object file. Next. Then there are 2 ways to access the content of ANSEP. Users can either create a free account into the platform and log in, or explore a limited part of the platform by accessing as a guest user. The major difference between a guest and a registered user is a personal storage space that the platform assigns to registered users that allows to upload files to be used in the analysis, and the possibility of saving the configuration and the results of performed analysis. The next. In summary, the future of the system-based approach is based on the ability to identify and evaluate the state of disturbed networks, thus allowing decision-making regarding appropriate interventions. Knowledge of the dynamics of the network and its interaction will allow us to advance in the understanding of the pathology based on the study of its interaction, as well as open a door to a specific modification of key subnets to prevent the evolution of the pathology. So in this moment, we can ask in the short to medium term, the integration of omics and associated models of disease will enable us to have a clearer picture of health and disease. So the answer is in our hands, right? The next. Finally, I want to thank to this great team of researchers, students, without whom none of this would have been possible. I thank Limerick University, Universidad Nacional de Colombia, and Mis Ciencias, Universidad Autónoma de Chile. I thank to George Barreto, Andrés Pinzón, Andrés Aristizábal, David Ramirez, on all this bunch of people, the great team that is working in the computational and structural lab at Pontificia Universidad Javeriana. Thank you for being here today, and I really appreciate that you took the time to be here and listen to my presentation.
Thank you, Janneth, for your nice talk. So we have time for questions. Someone has any question? So Janneth, the platform that you showed us is already available?
Oh yeah, I can send you by chat the link.
Yeah, please.
For you to know it, yeah.
Yeah, okay, thank you. So Wendy, are you here?
Yes, I'm here, and we will start with the lightning talks.
Also, we have a question from Bupati.
Yeah, can you hear me?
Yes.
Oh, yeah.
Yeah. Actually, I was joined your presentation bit, not in fully observe your presentation, but you show the slide, design the drug molecules for neurodegenerative disorder, right?
Oh, so sorry, I'm not sure that I understand the question.
You design the drug, actually, I never asked the question still. One slide, you show that design the drugs to neurodegenerative disorder, right?
Oh, you present on a slide with neurodegenerative disorders.
Disorder, right?
Yeah.
Yeah. Actually, in the neurodegenerative disorder, it has a number of neurodegenerative disorder. For example, Alzheimer's disease and amyotrophic lateral sclerosis and some other Parkinson's disease so far.
So you mentioned a group of diseases.
Yes. Can you focus which disease you are designing the drug to inhibit some amyloid protein aggregations? Can you got my point?
If in these diseases you find the amyloid protein.
Yeah. Which drug is? Can you find a drug for which neurodegenerative disorder? That is my main question.
Yeah, if you find some drugs for these disorders.
Find the drug for which neurodegenerative disorder?
For what neurodegenerative disorder you find a drug?
Yeah.
Oh no, not yet. Oh no, not yet. But we are focusing on 5 new targets and try to understand in an holistic way all the molecular mechanisms driving diseases. But
So
we are so far to find a new drug.
Which mechanism you are investigating or illustrating?
So sorry.
Which mechanism?
Yeah.
Which of the mechanisms were?
Yeah.
We have a lot of them, but we are focusing specifically in the fatty acid metabolism. So we found that the increase of cholesterol and all the effects of palmitoic acid are very important in the development of neurodegenerative process. So we are trying, using different profiles, omics profile, metabolic, transcriptomic, and proteomic profile, trying to conciliate all the mechanisms that we can see with the result of the analysis of all these profiles. But one important issue is all the metabolism of fatty acids and cholesterol.
Okay, Janneth, thank you for replaying this question. So with this talk, we finish the first part of this session, and thanks all of you for coming. And